we are going to look at the cytoskeleton. Cyto means cell. Cyto means the cytoplasm. Okay, it comes from the cytoplasm, cyto. Then skeleton is the skeleton, the framework, like a framework. A framework. So why do we need to look at the cytoskeleton? Cytoskeleton is very important for survival of our cells. If it were not there, the cells would not survive as they are able to survive. So how can we present the cytoskeleton? Let's say this is our cell. This is our cell. All over the cell, we have the cytoskeleton everywhere. So if this is our cell, that is our cytoskeleton. It crosses into the cell, into the cell, into the cell. That is the cytoskeleton. But again, our cytoskeleton has three basic components in our cytoskeleton. We have the first filaments. The first filaments up there. And these filaments are called the microfilaments. Then we have the intermediates. The next component are the intermediate filaments. Then finally, we have the microtubules. All these are part of the cytoskeleton. So cytoskeleton, the skeleton of the cell, we have microfilaments, intermediate filaments, then we have the microtubules. So, but the microfilaments are the smallest. Okay, they are if I'm to say they are the thinnest filaments within the cell, and they have a thickness or a diameter of about 7 to 8 nanometers. 7 to 8 nanometers, that is the diameter. Then the intermediate filaments, about 10 nanometers. Then the microtubules, about 25 nanometers. So these ones have the widest diameter. And these ones are kind of hollow as compared to the two divisions of the cytoskeleton. So what are we interested in? To dive into the details of each of the filaments. So the microfilaments, functions of the microfilaments. Microfilaments, functions. Microfilaments, again, what is important to note is that they contain what is called actin. We know actin is used in muscle contraction. So these filaments, actin coupled with myosin, will have muscle contraction. So actin, so number one function of the microfilaments implies that is, we'll need them in muscle contraction. The microfilaments, very important in muscle contraction. Now, what else can we use them for? Imagine we are having a white blood cell, and it is coming across a bacterial cell. Let's say this is a bacterial cell, and we have to engulf this bacterial cell. Let's say this is a bacterial cell, this is a white blood cell. It has to engulf. It will need to use the microfilaments for engulfing. So, and this is what we call phagocytosis. So in order for phagocytosis to happen, we need the microfilaments to form the pseudopod. This is what we call the pseudopod or the food for engulfing. Pseudopod or the food in our white blood cell, or if we have an amoeba. So that is phagocytosis. Then guess what? If we have a white blood cell still, and it wants to squeeze somewhere, to approach maybe a microbe that is in the body, we also need the microfilaments to take part in the activity. So, and that process is called diapedesis. So we need these ones in diapedesis. So diapedesis also, the squeezing of the white blood cell somewhere requires the microfilaments. So what else can we use the microfilaments?
let's imagine we are having a cell dividing,
cell division, and we reach the final stage, which is telophase, where we form the cleavage furrow. Let's say this is our cleavage furrow. Cleavage furrow. So the cell is going to divide. We have, let's say, a nucleus that side, a nucleus that side. So this process is what we call cytokinesis. 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 So also this one is aided by the microfilaments. So these are the basic functions, the functions of the microfilaments. We have muscle contraction, actin myosin, because they are composed of actin. Then phagocytosis, which is engulfing that microbe or anything. Then diapedesis, squeeze. Then cytokinesis, where we have formation of the cleavage furrow during cell division. We want to divide the cell into two equal cells or into two equal portions during cell division. So those are the microfilaments. Then intermediate filaments. So for our intermediate filaments, intermediate filaments. So for the intermediate filaments, I will start by giving examples of the intermediate filament. Remember, we said they are around 10 nanometers, so they have a larger diameters as compared to the microfilaments. So intermediate filaments examples, we can have like the lamins, the nuclear lamins. We talked about the lamins in our video of the nucleus, and we said the inner membrane of the cell envelope is surrounded by these lamins. And the mutation of the lamins leads to progeria, that condition. A mutation of the lamins leads to progeria. So lamins are one of the examples of the, and they are found in the nuclear envelope, the inner membrane formed by the lamins. Then we also have another type of intermediate filaments. We call them vimentins. Vimentins. These ones are found in the muscle cells. So they are supporting the muscle cells. Then intermediate filaments still, importantly, we have the keratin. So keratins are found in the epithelial lining of our skin, and they are also found in hair. In hair structures like nails. So they support the epithelial tissue, the keratins, a type of an intermediate filament. Very important. Then we also have another intermediate filament that is found in the neurons. Those are the new. Okay, we can call them these ones are the neurons, the neurofilaments. You can call them the neurofilaments. They are found in the neurons, stabilizing the neurons. So, but again, what we need to note about the intermediate filaments is that, to sum it all up, they can join a cell to cell, or they can join a cell to cell. We can have a cell to cell joint using these filaments. We can have a cell to extracellular matrix. Cell to cell to extracellular matrix. Then we can have cell to cell organelle. So cell to cell organelles are also joined. So we are having our cell A to cell B. We'll see this in cell communication. Then we have a cell to extracellular matrix, this cell to the extracellular matrix. Then we are having organelles inside here to cell. If this cytoskeleton was not there, the organelles wouldn't gain somewhere where they would be suspended very well. So we shall have the cell, like the nuclear lamins, we say they are used for stabilizing the nucleus. So cell to extracellular matrix, cell to organelles. Examples, neurofilaments for the neurons, then the keratins, the vimentins for the muscles, and the lamins for the nuclear membrane. So that is it with the intermediate filaments. Then finally we have the microtubules. Microtubules, we'll have a look at those ones. We say they have 25 nanometers. They are 25 nanometers thick. So we say they are hollow. They are hollow, they are tubes. They are, because 25 nanometers is at least much more larger than the 7 to 8 nanometers we saw in microfilaments, and the 10 nanometers we had a look at in the intermediate filaments. So, microtubules. Microtubules, basic function, cell division. Cell division. So we know them for cell division, separation of the chromosome, we break that chromosome, separation of the chromosome, let's say during cell division. This is what happens, the microtubules. Then we need to separate the chromosome. If we want to separate the chromosome, it will be done by the pulling action of these microtubules. So they are involved in cell division. Then microtubules again, another function with the microtubules. What can we think of the microtubules? Formation of cell extensions. Cell extensions. Cell extensions. What are the cell extensions? Cilia and flagella. Cilia and the flagellum. The cilia and the flagellum. These are the cell extensions. You can check out our videos for the prokaryotic cell, where we give the main divisions or the composition of the prokaryotic cell. We had a region of the cilia and flagella, the locomotory structure. So, cilia and flagellum can be used for propulsion. Flagellum for propulsion, like in the sperm cell, propulsion. Then the cilia, cilia can be used maybe for trapping dust in the respiratory system. It can trap dust in the respiratory system. It's beating action of the mucus such that it reaches regions where it is needed. Then cilia can also be important where? Can be very important in the female reproductive system for movement of the ovary. The cilia is used for beating the ovum to move towards where fertilization is going to take place, along the fallopian tube. So that is with the cilia. So that is it with the microtubules. Cell division, cell extensions like cilia and flagellum. Then also one important function. One important function with microtubules, they're used for transportation of organelles within the cell, of different particles within the cell. Let's say we have a vesicle and we need to transport the vesicle from one destination to another. We need the microtubules to do that transportation. So this is it basically with our cytoskeleton. The microfilaments, the intermediate filaments, and the microtubules. Size ranges, size differs, 7 to 8 nanometers microfilaments, 10 nanometers intermediate, and 25. Then the functions as we've listed. So that is it with the cytoskeleton.